Hello, everybody. Good morning. Good morning, Group 3. Our topic today is about heredity and hereditary traits. We will complete our lesson in the next session. We talked about the hereditary traits and acquired traits, recessive, dominant, and recessive. We compare and differentiate between Mendel's first law and Mendel's second law. Who was Mendel? Mendel is a scientist who is founder of heredity because he do experiments, a lot of experiments to identify the meaning of heredity and different types of hereditary traits. What is the difference between Mendel's first law and Mendel's second law? Mendel's first law, it has one pair of contrasting traits, maybe long and short, green and yellow the color seeds of pea plant. But Mendel's second law, two pairs or more contrasting or opposite traits, like long green and yellow and short yellow. So we have two types of contrasting traits. We will review quickly about the dominant and recessive traits of the human being, and we will study, inshallah, the gene composition, the chemical structure of chromosomes, and identify how the hereditary traits transmitted from one generation to another. Let's start. Okay, we have dominant and recessive traits of the human being. For example, hair, curly and black hair is a dominant trait. Smooth and light color hair is recessive. Eye, the white eye is dominant trait. The narrow eye is recessive. Dimples, having dimples, dominant traits. No dimples is a recessive trait. Freckles, no freckles, dominant trait. Freckles is recessive traits. They are opposite to each other. Ability to roll your tongue is a dominant. Inability to roll your tongue is a recessive. Free ear lobe, free, not attached, it's a dominant. But attached earlobe will be a recessive trait. Eye color, black eye color, dominant. And colored eye, blue, green, gray, brown, and so on. It's a recessive trait. It not appears in the first generation, but appears in the second generation. Okay, that's easy. Now we will study the chemical structure of nucleic acid, DNA. What's a chemical structure? Chemical structure of DNA, chromosomes consist of DNA and protein. DNA, which we will study its structure in details later, inshallah, in secondary one biology. But in this year, we focus only on the composition of DNA related to the genes and protein. The genes, which is responsible for transferring the hereditary traits from one generation to another. Let's see how we transfer. Let's see how can the gene transfer the hereditary traits from one generation to another. The genes gives a specific enzyme. This enzyme, which is responsible for making chemical reactions, like enzymes in your stomach or in your intestine and so on. salivary glands, gastric juice, bile juice. These enzymes make certain chemical reaction to produce protein. What's the function of protein? Protein showing a specific hereditary traits. Proteins is responsible for showing the specific hereditary traits like the color of eye, color of hair, earlobe, and so on. Okay, is it easy? So we have inside our chromosomes DNA, which contains gene. Gene is responsible for producing or giving specific enzyme, which is responsible for certain chemical reaction that produce protein and produce gives specific hereditary traits. This is the simple meaning or the simple function of gene and the simple meaning of how the hereditary traits transmitted from one generation to another. We have two applications for the heredity. First application is the genetic engineering biotechnology. Genetically modified rice with carotene. What are the genetically modified rice? I can modify the genes of any substance. Yes, we can insert certain gene which is responsible for inserting specific enzymes which gives certain protein that gives a specific hereditary traits. So the normal rice has the white color. The modified rice with carotene have orange color, like carrot. So because carrot has the carotene substance, which is responsible for producing vitamin A. So some scientists modify the rice by inserting the gene of carotene. This carotene genes result in creation of the provitamin A compound inside the tissue that stores the starch in the rice grain, which give the rice a lot of starch and it will be more healthy. So I can insert a specific gene which produces a specific hereditary traits inside the substance, especially in plant. We can modify the properties of plants according to the inserted genes. Okay, is it easy? Another application will be the Human Genome Project. What's the meaning of the Human Genome Project? It's a genetic map like maps in social study. It's a map that shows the complete set of genes that are present on the human chromosomes. We studied the chemical structure of chromosomes consists of genes, contains DNA and protein. Okay, so Human Genome Project is a map consists of a lot of genes or all of genes inside our bodies. This result of this project, the project shows the similarity of humans is more than 99%. As we are in the same speech, we are human speech or especially mammal speech. We are similar in 99% of characters, but only 1% has different characters. So the only 1% differences in humans, such as color of eye, color of skin, height, and other traits. All of them are only 1%. But more than 99%, we are similar, okay, as we are in the same speech. The Human Genome Project, this map, um, like our chromosomes in our body. They are arranged to be similar or different. This color show the differences and similarities in the chromosomes. DNA of the molecules, I told you, you will study it later in second year biology, inshallah. This information is so important. We have 46 human chromosomes in our body, 23 from male and 23 from female. So you most like your parents, but not identical to your father or identical to your mother, because you have only half of characters of your father and half characters of your father. Okay, is that easy? We have finished our lesson. Thank you for listening, but we have a lot of things to do in our session. I want to show you a video. This video is application of heredity. We have the main hereditary or very important hereditary traits in transmitted from one generation to another. It's called blood group. The type of your blood group is related to your father and mother. We have four types, but in fact, we have only two types, A or B. We have anti-agent A or anti-agent B. If the blood coagulated with A, so your blood group is A. If your blood group coagulated to with B, so your blood group type is B. If your blood coagulated with A and B, so your blood group type is AB. If your blood don't coagulate neither A nor B, so your blood group type is O. Okay. Anti-agent D related to the RH. Most common of our bodies have RH positive, but some people have RH negative. Okay, let's see our video and to recognize more what has happened. Okay, let's start.
Your blood group at home. These are three
Let's focus in this video to recognize well what is the difference between the four types of blood groups.
On your finger, this is my finger, and I'm making a prick with the needle, and then 3 drops of blood is being placed on the slide like this. And after that, we will put.
I will ask you after finishing this video about the difference between the four types of blood group. Please focus.
So, in the first, we will use NT.
This is anti-agent D,
NTB,
which are related to RH.
The NTD is being put. Now, 1 drop of NTB is being put in the center.
It's anti-agent B.
Color. And now, the blue color is NTA.
It's anti-agent B. Anti.
Right.
D, anti-A, anti-B.
And with the 4 corner of the slide, we will just mix.
We'll mix this substance with your blood to know if your blood coagulated with it or not.
See there for about 2 minutes, antigen antibody reaction will happen, and you will see it in the form of agglutination reaction. Agglutinin will come. Just do not mix it from the same side. I am using it from the other side of the finger. Sorry, another side of that. You can see the needle cap.
After time, for 2 minutes only, you will see the difference in the shape of the blood.
Just wait for 2 minutes, it's a little bit shaky, and then properly you will see a.
So after time,
splitting.
The blood will coagulate it with B.
How the B negative.
It's negative like. Because it's not coagulated with RH.
The B positive looks like this. The O positive is like this. The O negative, the AB positive, and the AB negative.
This is all the types of your blood group.
Thank you for giving your valuable time. And
okay. It's so interesting activity. I will do with you in our science lab later, inshallah. And now we will solve page 65 in your booklet. Please open your booklet, worksheet 7, page 65. Let's answer this page. The first question, write the scientific term. Traits that are transmitted from one generation to another, called hereditary traits. Number two, the traits that are not transmitted from one generation to another, it's called acquired traits. Number three, the scientist who placed the basis of scientific studies of heredity. The founder of heredity is called Mendel scientist. Please focus and differentiate between number 4 and number 5. Sorry. If two individuals that are different in a pair of alternative or contrasting traits couple, only the dominant characteristics appear in the first generation, while the two characteristics appear in the second generation at a ratio 3 to 1. This is the definition of the first, Mendel's first law. If the two individuals that are different in a pair or more of alternative or contrasting traits, the traits of each pair inherited independently and appears in the second generation of the ratio 3 to 1 or 9 to 3 to 3 to 1. It's a Mendel's second law. Okay, you know the difference between the first and second laws of Mendel. After that, question 2, explain why. Mendel chose the pea plant to conduct his research. Why Mendel chose the pea plant? It's the first thing we had studied in this lesson. Pea plant have short life cycle. Hermaphrodite or bisexual plant can be self-pollinated, give a lot of plenty of crops. In this question, you can write 2 or 3 points only. It's enough. Number 2, the free ear lobe is dominant. Sorry. Over the attached earlobe. Why this free is dominant and attached is recessive? Dominant as it appears in the first generation, while attached recessive because it can't appear in the first generation, but it appears in the second generation. Is that easy? Okay. Please solve page 66, 67, and 68 as a homework, and I will check this homework with you in the next session, inshallah. Please study well. It's so interesting lesson in your curriculum. We had finished our lesson, and we have only one lesson later, hormones. We'll study it in the next session, inshallah. Thanks a lot for your listening. Have a nice day, and I will meet you sooner, inshallah. Goodbye, my dear students. Bye.